Welcome to the second module of this course. My name is Barbara Mannweiler. I'm a project manager at the Institute of Translational Research. And in today's lecture, we will focus on phrasing a clinical trial objective and choosing the appropriate trial design, asking the right questions in the right way. To begin, we will have a very brief overview over the different stages that we have to go through when it comes to clinical research. First, there is, of course, basic research, which you are probably very familiar with. And from there, we can have the chance to discover potential drugs. These are usually around 5 to 20,000 compounds that have the potential to, at one point, get marketing authorization and go into a person. From there, we have the preclinical research phase where only around 250 compounds enter. So this is the stage that is immediately leading to the clinical phase. And you can already see the huge reduction of possible candidates in this stage. And from there, only 1 to 5 compounds have a chance to actually enter the clinical research phase, where it comes to trials of phase 1 to 3, which we will have a look at in a few slides in more detail. So you see that from the 5 to 20,000 compounds, only around 5 can make it to the point where they have a chance to get a marketing authorization. And even afterwards, we have further studies which have to be done or can be done, which are so-called post-marketing or phase 4 studies. And we will now go through all of them in more detail so you get an understanding when you want to design your own study, where do you have to place it in this whole construct, and what is the actual objective that you have to consider when planning such a study. So we go back to the preclinical stage, where we are doing in vitro and animal studies to investigate the absorption, distribution, metabolization, and elimination characteristics of the possible drug compounds. And of course, we do toxicity studies to see how the compound interacts with an organism, either in vitro or animal study, and to just get an idea about the actual safety profile. And as you can imagine, at this stage, a lot of compounds will already be dropping off because the toxicity profile and the general interaction with the organism will not be favorable. But if they are, and if they manage to go through the preclinical research phase, we will enter, after the preclinical phase, the actual clinical phase, which is our main topic for today. And this clinical phase is made up of different phases, which are consecutive. So we have first to do a phase 1 trial, followed by a phase 2 trial, a phase 3 trial. And if you remember, in the slide that we have just seen, this is the point where marketing authorization can be obtained. And afterwards, we will proceed with more phase 4 studies. So now you know that there are like a lot of stages that you have to go through when you want to like proceed with the drug development, and you have to do clinical trials. But what actually is a clinical trial? And to answer this, we will have a look at the current EU Clinical Trials Regulation, or short CTR, which describes what we have to consider when planning these clinical trials. It gives the regulatory basis for all the planning and also defines what is actually in scope for this regulation. And according to this, a clinical trial is a clinical study which meets the following criteria. First, the assignment of the subject to a particular therapeutic strategy is decided in advance and does not fall within normal clinical practice. Additionally, the decision to prescribe an investigational medicinal product is taken together with the decision to include the subject in the clinical trial that you're planning to do. Or diagnostic or monitoring procedures are done in addition to the normal clinical practice. So if you're like doing an additional blood draw or an additional CT scan, this would be in scope for a clinical trial. And of course, if you are planning an investigational, to use an investigational medicinal product in your study, and the decision to give it to a patient and include him goes together, it is a clinical trial. And according to CTR, all other clinical studies that are not a clinical trial are non-interventional studies. And what that means, we will have a look at a bit later in this topic. So let us proceed to the actual phases. So now that you have an understanding of what falls under a clinical trial, let us see what the actual objectives are that you have to tackle with your clinical trials. With a phase 1 clinical trial, the objective is to investigate the safety of the compound. So we are doing single or multiple ascending dose studies, and we want to get information about the pharmacodynamics of our drug compound, which is the biochemical and physiological effects of drugs and mechanisms of their action. And we need to gather information about the pharmacokinetics, which is the process of how a drug is absorbed, distributed, metabolized, and eliminated by the body. And you may remember these details from the preclinical phase, where we also had a look at these things. But here we are for the first time in the clinical phase. So we are for the first time in the human body. And of course, we have to have a closer look at all these characteristics again when we're not in vitro or in vivo or in any other study than human. So this is very important to, again, observe. The participants in phase 1 studies are usually healthy volunteers. Depending on the indication and the disease area that you are investigating, it may be different and not be done in healthy volunteers, but that heavily depends on what you're actually researching, and usually as healthy volunteers. The number of included participants is very low, so we are only in the range of double-digit numbers, and therefore it's done mostly in a single trial site because there's like this no number of participants. And therefore, phase 1 trials are also called first in human because it's directly following the preclinical phase. It's the first time we ever bring this drug compound in the human. So this is called first in human study. Following phase 1, if we were successful and everything is still looking promising, we can enter phase 2 trial. And the objective of a new phase 2 trial that follows the first phase is to find the right dose in terms of efficacy. So when do we see the desired effect? And of course, and as always, we want to detect the important safety signals. Now we include patients into the study, and the sample size is now high double-digit to hundreds of people. So we are now able to detect important safety signals, which may have been invisible in the very few persons that we investigate in a phase 1 trial. If it's like super intense, we may also see it in phase 1 trials, but with hundreds of people, we have a chance to now detect very important safety signals. This is done usually multi-center because we have a lot more people that need to be included in the trial. So we want to have more resources for recruitment of the people. And this is then called first in patient study. So phase 1, first in human, in usually healthy persons. And phase 2 trial is then first in patient because we want to see right dose in terms of efficacy. So we need to do this in patients, of course. And to make this a bit more like clear to you, I want to present you one of our own studies, which was also a phase 2 trial called a multinational phase 2 randomized adaptive protocol to evaluate immunogenicity and reactogenicity of different COVID-19 vaccine administration in older adults already vaccinated against SARS-CoV-2. And to break it down a bit, we will go through the different aspects of this phase 2 trial. It was a randomized trial focusing on participants of advanced age, so 75 years and plus. And it is a head-to-head trial comparing 2 different mRNA vaccines, which were both non-variant adapted, as the first booster vaccination. So the elderly people that were included in the study, which were 2 times 300 subjects, and therefore like a representative number of what we have seen on the previous slide. They have already been vaccinated 2 times with a COVID-19 vaccine and now receive one of those as their first booster vaccination. And the question behind this that we want to answer in such a trial is to see if and how this population actually profits from a booster vaccination, because at the time there is no information on this question for elderly people. And the important step when you want to answer such a question and try to design your trial is that you find the right objective, which is I want to compare the immune response between treatment arms after third vaccination doses against SARS-CoV-2. So this is actually what we want to, the question that we want to answer. And for this, we have to define a proper endpoint, which is in this case, the rate of twofold antibody titer increase following third dose, measured by a specific procedure that we defined. And this is the important step that you need to go through when you plan your trial. You have your research interest, you have a question you want to answer, and then you need to define what people do and need for this, how can I get them, and most importantly, what is my objective and what is the endpoint I can use to get an answer to my objective. So this was just to show you an example of how such a trial design could look like for a phase 2 trial. And especially for phase 2 trial, depending on how complex they are and the endpoint and objective that you defined, it may be needed to further subdivide phase 2 trials, which is very often done in the following ways. So especially phase 2 trials are subdivided into phase 2A trials, which are used as a proof of concept trial. So we just want to see if the intended effect of our drug compound can actually be detected. So is there the expected effect that we wanted to see at all? And in the second step, it follows the phase 2B trial, which is the dose finding trial. So after we have seen that we can trigger the effect, we want to find out the perfect dose to administer. And therefore, we have a look at the impact of different doses on the response parameters and use these data to define the perfect dose to be used in the further trials, and which will ultimately probably also go into the marketing authorization. So it may be useful, depending on complexity and what you want to see, to split these phase trials in a proof of concept and then a dose finding part. And if you've successfully done that, you're able to enter with your compound into a phase 3 clinical trial, which you again have to, of course, newly develop. But this is now done with the objective to get a proof of efficacy and safety, which we have also done in the previous studies. But now in phase 3, we do this in comparison to the standard therapy or placebo. Of course, again, it depends on your research question whether a standard therapy or a placebo should be chosen. That is super individual and there's no one-size-fits-all procedure. But as always, we want to see it's effective, we want to see it's safe, but now in comparison to what we already have or to nothing, of course. Again, phase 3 trials are done with participants, of course, because we want to see about the efficacy in comparison, especially to standard therapy. We need the patients. And now we enter a range of really huge numbers from high triple digit to thousands of people that can be included into phase 3 trials. And therefore, the study duration is also absolutely variable and it depends on all the interventions that you're planning, on the disease you're investigating. So if it's very slowly progressing, you need a long time with probably a lot of study visits to actually see the changes and see the efficacy of your drug compound. And you may also be in need of longer follow-up periods, especially if it's a question of flare-ups again that will happen at a later time. So again, this is super individual, but it may range from days, weeks to years that you have to perform such a study. And due to the high number of patients to be included, This is very often done in multi-center trials, so you use as many trial sites as you need to get to the number of patients that you need. And it's also even done multinational. So you do not only use the resources in your own country, but use also the clinical cases abroad. And this is then called a pivotal trial, which means it is a study to generate safety and efficacy data that are the decisive basis for the marketing approval application. Because as you remember from this oversight, if we successfully go through a phase 3 clinical trial, this can give us the data that are needed to proceed with the marketing authorization. And this is the critical phase where you need to obtain these data. Of course, data from all the trials and probably also from a lot of phase 3 trials had to go into that. But at this point, there may be a chance to get the marketing authorization for your drug compound. But as you have also seen, the number from 20,000 to only 1 to 5 compounds is decreasing in such an enormous way. And as you can probably imagine, there have to be some major factors that lead to this decrease. And if you remember back to what we heard about all the objectives of the different phases, we are always talking about efficiency and safety, of course. So the main reasons why compounds do never reach the phase of a marketing authorization is usually a lack of efficacy, so no efficacy at all, or not sufficient efficacy. So if we have like a negative risk-benefit ratio, the safety profile, toxicity profile is not in favor and does not outweigh, or the efficacy does not outweigh the safety concerns, then there's no way this compound will proceed in the clinical development. So either we see no efficacy or we have an unfavorable safety profile. These are the most common reasons why the drug compounds fail in the clinical development. Unfortunately, of course, because like with every compound, you have all the hopes and you proceed with the most promising, but it may still be seen that they just cannot proceed in the clinical phases because of these issues. But regardless, we just hope that we have received all the data with great efficacy and very low safety concerns so that we get our marketing approval. And still the road does not end here because as you have seen previously, we also have post-marketing studies, which are the phase 4 clinical trials. They all happen after the marketing authorization. And the objective here is to like observe whatever your compound is doing under the everyday conditions. Because so far, as you also remember the definition of a clinical trial, we have a very narrow range of people that can be included in the phase 1 to 3 trials. We have a lot of restrictions on what we can do, what we observe, how long we observe it. It's very strict. And now in phase 4, with the marketing approval, it can be distributed to all the patients, just depending on comorbidities and just prescription of the physician that is giving it out to the patients. But we have the chance to now observe our compound under everyday conditions. So we get information about the long-term safety, rare and very rare side effects, because we now just have like so many people that are taking it. And we here have the chance, because it's marketed, to either create a clinical trial as a phase 4 study or do non-interventional studies. We will go back to that in a few slides, what the non-interventional study actually means. But now, after marketing approval, you're not bound to do a clinical trial anymore, depending on what you want to see. The participants, again, of course, are patients which are eligible to receive the marketed drug. And now we are talking about thousands to tens of thousands of people, which allow us to get all the information that we just talked about regarding safety, side effects, which is really a great way to see what the compound actually does in the non-restricted environment and actually see what it does to the general population. So these are the post-marketing studies, as I just said. Usually there are like a few different types of studies that can be done or have to be done after market launch, which is for one, the post-authorization safety study or the post-authorization efficacy study. They, as I just said, can be done either as a clinical trial or a non-interventional study. You already knew now about the clinical trials, which were like very narrow and regulated. And on the other hand, we can do the non-interventional studies, where the trial design is usually a cohort study or a case control study or a registry. Just to give you some examples, there are a lot of different ways that you can also create your non-interventional study. And again, you just have to ask yourself what you want to see, what you want to, or what question you want to answer with your trial. And we will now just go through all the study types that we could do after marketing authorization. Firstly, there is the post-authorization safety study, where the purpose is to evaluate the safety and risk-benefit profile of a medicine. So we have the market authorization, it goes to thousands of people. And in this phase 4 environment, we can just do a study like that to see more details about safety and risk-benefit. We can, with this, collect further information on the medicine safety. And on the other hand, measure also the effectiveness of the risk management measures that the authorization holder put in place. Because, of course, things can happen, adverse events can happen, but there needs to be a risk management system in place. And with these studies, it can just be proven that this is working properly. On the other side, there are the post-authorization efficacy studies, where the purpose is to evaluate the efficacy. And this is especially necessary if there is still an uncertainty about sufficient efficacy at the time when you receive your marketing authorization. And for both of these trial types, it can be either of two ways. Either such a study, especially as you can see in the efficacy study part, when there are still uncertainties about sufficient efficacy or safety profile information, it can be imposed by regulatory authorities. So you have to do these studies after receiving the marketing authorization, or they can be done voluntarily by the authorization holder, of course. In any way, these studies may influence the actual marketing authorization status. So it can either remain as it was when it was issued, it may be changed, or it can even be revoked. So if you see that the efficacy in the broad population is way lower than in your very controlled trial environment, or there are suddenly a lot of side effects happening that you could not have seen before, that may lead to a change or even revoking of the market authorization. And as I've said previously, we can either do clinical trials or non-interventional studies already for this type. And now coming finally back to what non-interventional studies actually are, we for a moment say goodbye to the clinical trials and fully delve into non-interventional studies. And according to the clinical trial regulation, which gave us a definition of a clinical trial in the beginning of the presentation, we now see that according to this regulation, a non-interventional study is a clinical study which is not a clinical trial. So in order to understand this definition, you have to go back to see what actually is a clinical trial, where it was all about doing something that is not standard of care, it being done additionally, being involved in a clinical trial and provided with a medication that you wouldn't have received otherwise. And to help you understand what this now means, and not thinking just about the inverse of what you've seen several slides ago, we will just very quickly go back to the old definition of non-interventional studies coming from the Clinical Trials Directive. You may still see some studies following the clinical trials directive, but this is by now coming to an end because this directive has been superseded by the regulation. But still, it helps you understand the difference between a clinical trial and a non-interventional study. So according to the superseded definition, a non-interventional study is a study where the medicinal product is prescribed in the usual manner and in accordance with the terms of the marketing authorization. The assignment of a patient to a particular strategy is not decided in advance by a trial protocol, but just falls within the current practice, and the prescription of the medicine is clearly separated from the decision to include the patient in the study. And lastly, no additional diagnostic or monitoring procedures are applied to a patient. So if you just try to remember the definition of a clinical trial according to CTR, you actually see that it's just the inverse of this definition. So it's not as superseded as you think here. It's just made a bit easier by the simple sentence, but yeah, so if you just follow standard of care and do no additional interventions, there are good chances that what you want to do is a non-interventional study. And as you've previously very briefly seen, there are like a few different major types of non-interventional studies that I want to present you. So this is like the cohort study, which is usually done in a prospective way, a case control study, which is retrospective procedure, or a registry, which is also a retrospective approach. A cohort study, as I've just said, is mostly prospective, and the study groups, or the cohorts, as the name indicates, are characterized by different baseline criteria or treatments. And these cohorts are then just observed over a longer period of time for frequent conditions, and you can just draw your conclusion from these observations. This is ideally done for phase 4 safety or efficacy studies that we have just learned about. So you just generate your cohorts, observe them, and see what happens. But you need to remember, if this is not done as a clinical trial but non-interventional study, you are not allowed to do any interventions that are not routine. So no additional blood draw, no additional CT. You have to stick to the clinical routine for this. In a case-controlled study, as the name suggests, the starting point is an already existing event, so the actual case. And from this existing event, we go back in time and attempt to determine the risk factors that led to this event happening in the future. So we compare the cases which have the event and the controls that do not have this event. And this is mostly used for rare diseases because we don't have that huge pool of possible study participants because it's a rare disease. So it may be very helpful to just look at the individual cases and go from the event back to the factors that led to this event happening. So in sum, it's matching the cases and the controls, which is very important here to have the proper comparison to draw proper conclusions. Lastly, in a registry, as you may probably already know, it is a retrospective documentation of individual cases, but of a defined disease. And to give you an example on how a registry works, we will just go through an example to make it a bit more easy. And this example is also one of our own registries, the so-called FungiScope registry, which is a global registry for invasive fungal infections. The objective of this registry is to collect epidemiological and clinical data on invasive fungal infections. So we use a web-based eCRF, which is actually just an interface where a lot of clinical data and patient data can be entered. However, it is always anonymized and allows to retrospectively document whatever happened in that case, in this clinical case, and to assess the risk factors, get information about diagnostics, treatments, and deduce further outcomes and health economic impacts, for example. So With documenting these clinical cases, we are able to assess the different risk factors that led to this infection, and from there, try to develop treatment strategies and work on recommendations on how to treat certain patients with certain infections. And additionally to that, the Fungiscope registry also contains a biobank where the isolates go to so that species identification can be done, susceptibility testing. And this is a great way to just combine the individual clinical cases with the microbiological aspect. So we don't only have the data, but we also have the biomaterial, which allows for great collaborations with other groups for joint analysis, which especially in the fungi scope terms is also time and again used for marketing authorization data. So this is really a great and robust registry that can be used widely and, as it said, from various groups to generate the data that are needed to support certain developmental stages. The inclusion criterion for this study is an invasive, or not for this study, but for this registry, is that an invasive fungal infection has been documented by culture histology PCR. And with that, we were able to implement over 3,500 cases since 2003 with partners in 92 countries and 8 central laboratories worldwide. So we have a great registry with anonymized retrospective data, which based on the web-based interface is super easy to use worldwide and allows for worldwide collaboration. So with this, we come to the end of a part about the different phases and clinical trial types that you will encounter and may also plan to do yourself. But there is still the question of how do we actually plan such a clinical trial? What do we have to consider? So if you know at what stage you are, you probably by now have an idea about what phase you are in or what phases you have to go through, or if you're like just in the department of post-marketing studies, but still, we need to learn about what do you have to think about? What is your to-do list made of? So how was it planned and which tasks and to-dos do you need to consider when preparing, conducting, and finalizing a clinical trial? And of course, and obviously, it all starts with the idea that you probably already have. So starting with the idea, you can go ahead and plan your clinical trial. From there, start with the preparations, actually conducting it, complete it, and of course, analyze and publish what you have found. And we will now go through the different stages so that you can get an idea about what's behind all these different stages. So following the idea that you have, whatever you want to find out and whatever question you want to answer, the most important question that you have to ask are the actual research questions, where you can very easily follow the PICO process. So at first, you have to consider which patients can be included. So you have to define your population and what the inclusion and exclusion criteria are. So you have to decide what people are actually eligible to deliver the data that you need to answer the research question. In the second step, you have to ask what treatment should be investigated. So this will be the I and the C in PICO, the intervention and the control. So you have to develop the treatment strategy, what the drug that should be administered, in what way, in what dose, in what design type should it be administered, and what should be used as a control. So what do we compare it against? And lastly, you have to ask yourself in this very first step how the treatment outcome should be determined. So you have to define target variables, or so-called endpoints, that we have seen way back in the age study as an example for a phase 2 study. You have your research question, you know what you want to ask, and now you need to define a way to answer to that question. And therefore, you have to define primary endpoints, of course, which are clinically relevant and are actually able to answer the primary research question. They should be reliable and validated so that there is no doubt about whatever you find. And these are also the basis for the case number estimation, because once you know what you need to answer your question, you can also deduce how many patients or healthy volunteers at whatever stage you are, how many subjects you need in your trial to answer this question. Additionally, you can define secondary or even exploratory endpoints that can either support in terms of the primary research question or do answer some secondary or explorative research questions. Following that, as we have just very briefly mentioned in terms of case number estimation, you have to consider your statistical planning. So you have to probably look for some expert and do all the statistical assumptions, determine the type of outcome analysis, the randomization and blinding, if they should be included in your trial, which again depends on what you're actually looking at. So to make this a bit more understandable, I want to do a very quick excursion to randomization and blinding. And randomization is the process of assigning trial subjects to treatment or control groups using an element of chance to determine the assignments in order to reduce bias. So in simple terms, you just let, like flipping a coin, chance decide whether your patients receive treatment A or treatment B. And because you cannot influence what the person standing before you receives, of course, we reduce the bias in the treatment assignment because you cannot do anyone a favor, for example. To show you how this could look like in a very simple setup, we have the population of enrolled study participants, which are undergoing a randomization step, which means in this very simple case, half of them will receive the investigational medicinal product, follow on or go on with all the study procedures, trial visits until the endpoint is reached. And the other half of the group will receive the control treatment, will also go on with the trial until the endpoint is reached. Of course, this can be way more complex. That can be like initially randomization, and there can happen like all possible scenarios of how these two arms could proceed, or you can even have more arms. So again, it's super flexible. But this is a very simple setup to show you how randomization works. And blinding is a procedure in which one or more parties involved in the clinical trial are kept unaware of the treatment assignments. So this is also, like randomization, a very often used procedure in clinical trial planning or clinical trial conduct, that either single blinding is included, which usually refers to the subject being unaware of what they receive, or a double blinding, which refers to, on the one hand, the subject being unaware, and on the other hand, the investigator, monitor, and in some cases also the data analyst being unaware of the treatment assignment. And this is usually done to reduce a potential bias due to knowledge of the treatment assignment. So if no one knows what a patient receives, we cannot so the patient cannot be the patient will probably not be biased in how they feel about the medication that they are receiving. The practitioners will not be tempted to treat them differently because they know what they receive. And this is thus a very effective means to reduce the bias due to treatment assignment. But this is a very important point that you always have to consider if you want to do a blinded trial. There have to be robust procedures for emergency unblinding in place. Because at any given time, it can happen that there are very serious adverse events happening that may be related to the study treatment. So you have to immediately be aware of what a patient receives, although this trial is blinded, in order to provide the proper care for the affected patient. So this is something that you always have to consider when doing a blinding, that you need to have a proper emergency unblinding procedure in place. Further important point that you have to consider, but that unfortunately are still not exhaustive, are, for example, financial planning aspects. So if you want to do an investigator-initiated trial, do you need to apply for further funding? How is your budget structure? What do you need in terms of expenses? And how is this financed? You have to compile the clinical protocol, of course. So we at some points briefly mentioned the protocol. So you have to have this one document where everything you plan to do is written down, and this is the trial protocol. So this is fixed. This is one of the most important documents of your whole trial. So at a very early stage, you have to work on that one because it just defines what you're doing in your trial. Then you should think about the size that you want to include. How many of them do you need? Which of them have a suiting profession that you need, and so on. You need to think about the recruitment strategy. So how do you plan to get the patients into your trial that you need? And there's still a lot more that you need to consider. But the planning stage is very, very important because everything you're doing here in most detail will help you in the end. So it's really important that you take care and time here and that you may reach out to any specialist that can support you here because it's very important that a trial has a robust fundament. And after having everything planned and set out, you can go on with the preparation. So you can do contracts, or you have to do contracts with everyone involved, all the external parties like the trial sites or other vendors that may need to be included. You have to work on all the protocol, on all the documents for the study, like the protocol or patient information or consent forms. You have to work on the data capture platform. And very important is that a lot of those documents and strategies that you are planning have to go to ethics or regulatory authorities. So a very important step during the preparation of your study, once you have set out everything you want to do, is applying for an ethics opinion or approval and for the regulatory authority approval. So please, no matter how far you are in your planning and preparation, never start with a clinical trial without having the proper approvals or opinions in place. This is super important and can easily be supported by persons with very in-depth knowledge about the regulations that are applicable in your country, but always consider that this has to be done. And once you have everything in place and you are legally allowed to start the trial, the initiation visit can be done by a so-called clinical monitor, which will more or less allow the site to actually start recruiting the patients into your study. And with this, we go over to the conduct of the trial with all the study visits and the study interventions, the follow-ups. We collect all the actual data and perform, very important, the pharmacovigilance on an ongoing basis. So we get information about all the adverse events that may happen. And of course, constantly assess a risk-benefit ratio for the compound or whatever you are investigating in your trial. We do monitoring to ensure the data reliability and patient safety, and still a lot more. So this is like a leading theme. There's always more to consider. So we don't have any chance to go through all the details of clinical trial planning, preparation, conduct. In this, there's always a lot to consider, and probably also always more than you think your final to-do list counts. In the end, once all your patients have been treated, all the data have been collected, we near the completion of the clinical trial. So the monitor that I just mentioned will do a closeout at the visit, a closeout visit at the trial sites, meaning they will stop their work for your trial. The database will be cleaned and logged so that we have the final data that can be exported and analyzed. And this analysis will then ultimately go into a clinical study report, which is again sent to ethics and regulatory bodies, which entail all the actual study results that you generated. And afterwards, these results can be published. And that's when you finally reach the step that you wanted to have. You finalized your trial. You have all the information that you wanted to gather from that trial. You can go ahead, publish, and whatever your question was, you can proceed into the next phase, or you can like influence some recommendations. So this is the part where you have finally, reached your goal with your clinical trial. And as you have seen, there's a lot of steps to consider, so never hesitate to reach out to any professionals or specialists, especially. So anyone like being proficient in one of those single branches, because it will always help you in the end. The better you plan and prepare everything, and the more versed you are in how to conduct such a clinical trial, it would always give you the best data you can generate, which are very robust and will help you in the end. And because of this lot that you have to consider, the time from the idea in the very beginning to this point that we are at now with the analysis and the publication of results is again super individual. So we can give you no actual indication of how long such a trial will be or how long the whole preparation process will take, because this is highly depending on the phase of the clinical trial. So at what phase do you start with your work? What is the indication and disease area? If it's very slowly progressing, of course, we need a longer clinical trial overall compared to like very brief event that can be assessed, and then we go on to the next. So this is super individual. Also, the required number of subjects plays a crucial role, because the more people you need, probably the more time you need for recruitment. And of course, the more visits you plan into your study, the more time it takes. And ultimately, not only depending on the recruitment, but you also need to be aware of the enrollment dynamics of the trial site. So all this together heavily influences how fast you can get from an idea to your results. It may take like weeks, months to years, as we have said. So there's a lot to consider. There's a lot of things and aspects that could take time, but it's really a fun task to do. It's great to create such a study or such a trial. It's challenging, but it's also rewarding. So whenever you are in need of any further information, never hesitate to ask. So with this, I want to thank you for joining today into this presentation. And as I said, if there are any questions or remarks, please contact and not hesitate to contact academy@ucgm.de. Thank you.